It's a pleasure to have this opportunity to talk to you about our genomics work in Dresden and Okinawa. I've been involved in genomics for over 40 years now, and I thought I would take the occasion of my transition to an emeritus director to speak about this history in order to give a context of what is happening here today at this meeting. Amazingly, the core method of sequencing, the shotgun approach, has remained a constant during those 40 years, despite huge transitions in the technology for producing DNA sequencing reads. In this approach, a pure aliquot of a target, either genome or cloned piece of DNA, is randomly sheared, and a random subset of the resulting fragments are sequenced via some read sequencing technology. 30 years ago, the target was typically a 150,000 base pair back clone, and the read technology delivered about the first 500 base pairs or so of a shared fragment. Enough data was collected to typically cover the target 10 times or so. That is, in our running example, about 3,500 base pair reads totaling 1.5 million bases. The computational problem then is to build an assembler that reconstructs the sequence of the sampled target. This is a classic inversion problem. Given the result of a process, infer the source. To compound matters, the data is imperfect. Of paramount influence is the rate of error, call it epsilon, in the individual base calls in the reads, which was about 2 to 3% for the Sanger technology used 30 years ago. In addition, one does not know whether a read comes from the 5' or 3' strand of the target, i.e., a read's orientation is unknown. The biochemical steps in the production of the read can introduce untrimmed vector, ligated chimers, and foreign DNA, to name but a few read artifacts. In addition, the correct inversion becomes very difficult to infer in the presence of repeated sequences in the target. And finally, portions of the target may not be represented in the data set due to undersampling or sampling bias. So an assembler puts together reads that it is certain truly go together into contigs that represent contiguous intervals of the target. Given the redundancy provided by the oversampling, a consensus sequence of each contig is produced by multi-alignment that votes for the most likely base at each position. Note carefully that the result is not necessarily a single contiguous reconstruction of the target. Gaps occur either because there is no data or because the assembler could not resolve the region unambiguously. So the first shotgun project was the 48,000 base pair phage lambda at the start of the 80s by Fred Sanger and colleagues. The Sanger reads produced in this project were about 300 base pairs with an error rate of about 3%, increasing towards the end of the gel. I point at this paper because, in my opinion, it is a remarkable paper. In just 5 pages, 3 methods for read sampling are described, 4 methods for closing gaps are presented, and there is even a rudimentary program for assembling the data computationally. If you're going to read one paper from this era, this is the one to read, in my opinion. And in general, I think it serves as an example of what one should aspire to in their own work. It makes it clear why Sanger won 2 Nobel Prizes. So from my perspective, the first era in DNA sequencing, I term the Sanger era. The targets were all 150 kb or less, and the coverage of the target was typically 10 or less. The sequencing reads initially were about 300 base pairs, but length gradually increased over the years to about 700 base pairs on average by 2000. Depending on the machine and the method, error was about 2%, but increased significantly at the tail end. Basically, over the 20 years after the first Lambda project just mentioned, most of the improvements were in automation and sample cloning. In my view, the next big change occurred when the target went from being a 100,000 base pair clone to the whole genome, what I call the whole genome transition. So the belief around the 1990s was that the largest target that could be reconstructed via a shotgun assembler was about 150,000 base pairs, constituting a jigsaw puzzle of, 1,000 to 2,000 pieces. So to sequence, say, 150 million base pair chromosome, the Human Genome Project protocol was to build a physical map of 150,000 base pair back clones that were shotgunned from the chromosome. This physical map was effectively an assembly of the clones based on fingerprints, such as restriction maps of these clones. Given this map, one then selected a tiling set of clones that covered the target and shotgun sequenced each of these. So it was a secure plan in that it would produce a significant portion of the target. But there were some problems. The physical mapping was a completely separate process from shotgun sequencing. So you had to set up basically two distinct factories. The clones were unstable about 4% of the time. When you pull them out of the freezer, they were rearranged. The map itself was ambiguous, as the fingerprint information provides a weak inference of the overlap. In fact, the physical map of the human genome was never produced. And finally, and probably most importantly, one has to do thousands of shotgun preparations to complete such a project. So a counter to this prevailing intuition about the size of what could be shotgunned was the sequencing of the 1.8 million base pair H influenza genome at Tiger via Craig Venter's team, via a shotgun approach, basically followed by a gap closing strategy. Admittedly, this genome was not repetitive, so it was in some sense easy to assemble, but the feat nonetheless inspired Jim Weber and I to think about whole genome shotgun further, and in '96, we proposed that a genome the size and repetitiveness of the human genome could be well reconstructed with a shotgun approach that used paired reads, as I explained in the next slide. In the paired end protocol, the sheared fragments are carefully size selected, and then both ends of the fragment are sequenced, resulting in a pair of reads in opposite orientation that must be at an approximately known distance from each other in any reconstruction solution. The assembly is much as before, save that now the pairs between the reads link the contigs into potentially very long and oriented and ordered runs called scaffolds, where the size of the gaps between the contigs in the scaffolds is roughly known. This idea was not widely accepted, and critics abounded. But in 1998, Celera was formed with Craig Venter at its head to do exactly this, and to do exactly a paired end whole genome shotgun of the human genome, where the pairs would be produced at distances 2,000, 10,000, and 100,000 base pairs. This is the reaction to the formation of Celera as characterized by a New York Times cartoonist. But it was possible. The key to the method I developed was to understand what part of the genome was repetitive versus unique. This was affected by recognizing the parts of the problem where the reads go together in only one way and have the coverage signature of unique DNA. Technically, what I did was I found the uncontested minimal subgraphs in this graph of all read overlaps that had depth less than 1.4 times the coverage C or less. I call these subassembled parts unitigs. Given these single copy correctly assembled unitigs, the pairing relationships were then used to link them into long scaffolds with very high reliability. Lastly, many of the repeat-induced gaps between the unitigs could actually be backfilled by assembling the reads that had their mate anchored in an adjacent unitig. This worked as the repeat in the gap in isolation is often not internally repetitive. Altogether, this was a 20,000 CPU compute in 2000. So in the two and a half years, we produced the data and software to produce high quality drafts of not only the human genome, but also the fruit fly, mouse, and mosquito, proving the feasibility and efficiency of the paradigm, the whole genome shotgun approach. As a computer scientist, I was pleased that our expertise contributed to this accomplishment in a non-trivial way. So the next epoch in this short history, I term the short read era. Basically, the next generation of machines were all based on chain extension chemistry and thus limited to a read length of about 150 base pairs, but with error improved to 0.5 to 1%. To compensate for the shorter lengths of the reads, the shotgun projects typically involved a coverage of 30 or more. Celera had 300 Sanger sequencers. Over the last 20 years, the throughput of the next generation sequencers, the NGS sequencers, increased from 1/3 of Celera's output per machine in 2005 to 36 times Celera's output today. That's the equivalent of over 10,000 of the machines we had at Celera. Driving this era of short reads was the NIH's goal of the $1,000 human genome. Note carefully that the goal was not de novo reconstruction of an individual's genome, but mapping an individual's short read data to the consensus human genome to understand variation in the human pangenome. So the underlying computational problem was no longer assembly, but rather read mapping. So to me, this was a bit of a disappointing turn of events, and I basically left the field once I understood this. Specifically, the short read length meant that de novo reconstructions basically went to hell. Here you see the Contig N50 for the Celera fly reconstruction at upper left, and then the N50s for projects involving no pairing and or short reads. You can see the consensus, the N50 Contig length circled in a magenta oval in the slide. These are genomes, and these are genomes that are actually deposited at the NCBI. Interestingly, the world went from eschewing whole genome shotgun to performing it even when the data guaranteed a very fragmented reconstruction. So in this era, de novo genomes were really quite awful. They really were Swiss cheese. While one could still get interesting information and still do interesting biology with the data, the reconstructions entailed incomplete genes, missing paralogs and orthologs, entire regulatory regions were missing. You couldn't see the structural variants, and in general, the overall genomic structure. In short, a great deal was missing. Fortunately, around 2012, the third generation of machines, the long-read sequencers, began to mature, and the next era started. These machines produced very long but very noisy reads. So coverage was further increased to typically 60 to compensate for the increased error rate. The third generation machines are single molecule machines. And in the case of the PacBio instrument, they directly observed the incorporation of labeled nucleotides by an engineered polymerase, biotin affixed to the bottom of a zero mode waveguide. The reads were typically about 10 kb in 2012, gradually increasing with improvements to 35 to 50 kb today. But they have a very high error rate, 15%, heavily biased towards insertions due to the thermodynamic noise during the incorporation process. Initially, the community was unfavorable as this level of error noise was considered to render the data of too low a quality. However, unlike previous technology, read errors are truly random, being the result of this thermodynamic noise. And also, because they are single molecule machines, read sampling of the genome is also nearly random, as there is almost no bias in what finds its way into a well. These attributes of being random for these two facets of the read production process actually implies theoretically that these were the first technologies that could deliver unlimited quality simply by virtue of sequencing deeply enough. I tweeted my excitement about this in 2014. So indeed, when I arrived in Dresden to start my lab there in 2012, 2013, I decided to buy in on the PacBio long read instruments and set up a long read shop in Dresden. This facility, incidentally, has become the Dresden Concept Sequencing Center, which is one of 4 German excellence centers of the German Research Foundation. The initial projects that I undertook were the 70% AT-rich planarium that had defied all previous attempts at assembly, and the gigantic 32 gigabase pair transposon-rich genome of the axolotl. Both animals are important model organisms for studying regeneration. While not at today's standard per se, these initial projects produced reconstructions that far outstripped previous work at that time. From this early experience, I came to believe that high quality telomere to telomere reconstructions at a reasonable price was only a matter of time. So I joined others like Eric Jarvis, Richard Durbin, and Adam Philippi, who likewise saw the potential. And together we began to start sequencing vertebrates and bats in the nascent VGP and BAT1K projects. As a computer scientist, I personally was captivated by the idea that we might ultimately achieve perfect de novo reconstructions. So by 2016, we had agreed on a sequencing protocol that entailed collecting 50 to 60x of PacBio long reads and 3 types of scaffolding information, namely 10x read clouds, bionano restriction maps, and high-C read pairs.
For one billion base pair genome, assembling the long reads typically resulted in about 1,000 large contigs with an N50 in the range from 1 to 50, in the range from 1 to 5 million base pairs, and some detritus representing unassembled centromeres and other highly repetitive bits. Scaffolding then resulted in roughly 20 or so large scaffolds with an N50 of 40 to 50 megabase pairs typically, representing almost entire chromosomes or chromosome arms. The bar plot, courtesy of Arong Ri from 2019, shows the contig N50 and scaffold N50 in black and yellow, respectively, of the first 16 genomes produced by the VGP group. All passed our standard of a contig N50 of more than a megabase and a scaffold N50 of more than 10 megabases. And while not shown, all had a quality value of 40, that is 1 error in 10,000 base pairs in the consensus sequence. At a meeting at the Wellcome Trust in 2017, I presented this slide, estimating that with 150 million euro investment in 300 long-read sequencing machines, such as we had at Celera, one could produce 300 terabytes of high-quality genomic reconstructions per year at a run cost of about 225 million euro. With such a Celera-sized factory, one could sequence all 1,400 bats in 3 days at a cost of 2.4 million euro. One could sequence all 72,000 vertebrates in 9 months at a cost of 170 million euro. And one could sequence all 1.5 million known species of life in 15 years at a cost of less than 4 billion euros, which is less than the estimated cost of the Human Genome Project in inflation-adjusted terms. So this calculation shows that the cost of biodiversity genomics will not be the ultimate bottleneck. Rather, as is now clear to most, the issues of sample acquisition and preparation are the true limiters. That is, feeding a factory such as the one I outlined here is the problem. Finally, in 2018, a simple but clever shift in the running of the long-read instruments improved the error rate at a slight decrease in read length to an incredible 0.2%. Moreover, after homopolymer compression and a conservative correction step, the error rate for these hi-fi reads became effectively 0.01%. At this error rate and read length, reconstructions are phased and nearly telomere to telomere. So the key observation for hi-fi was that once the polymerase had made the first pass across the insert, subsequent passes were more processive and 2.5 times faster as the construct was now single-stranded. So by simply controlling the insert size more carefully and running the machine longer, 6 to 10 passes produced a consensus sequence for an insert that has only a 0.2% error rate. So 80% of the errors are homopolymer insertion or deletion. So by simply compressing homopolymer sequences, the error rate is reduced to 0.04%. And then a conservative voting strategy for correcting obvious errors reduces that further, typically about 0.01%. So in effect, one is working with near perfect reads. So the shift to hi-fi was not immediate, as there were cost concerns and the value of the shorter but very accurate reads was not fully appreciated. But today, the community consensus is to produce 30 to 40x of hi-fi reads, resulting in contigs with an N50 of over 10 megabase pairs, constituting 100 to 200 phased segments of an exemplary 1 billion base pair genome. Because the contigs are so big, it suffices to scaffold with just 60x high-C data, resulting in a single scaffold per haplotype phased chromosome. So the development of these really very high quality de novo genome has effectively enabled a rise of biodiversity genomics, the focus of this meeting. The VGP was one of the first initiatives begun at a meeting in a hotel next to Dulles Airport in 2015. Our center in Dresden has produced 20 of the 260 ordinal species at the first level of the project, totaling 42 billion base pairs of reconstructed sequence. At the same time, the BAT1K, led by Emma Teeling and Sonia Van Az, began. And to date, Dresden has sequenced 42 bats for this project, covering all the families of bats and totaling 84 billion base pairs of finished genomic sequence. A couple of years later, the Earth Biogenome Project began, and in some sense, this is an umbrella project for all the individual clay-directed efforts, such as the VGP, GIGA, and et cetera, et cetera. In what I consider to be an interesting tactic, the Wellcome Trust funded the Darwin Tree of Life project after the London meeting I mentioned earlier. The focus of DTOL on a geographic region as opposed to a clade of life was very strategic for two reasons. First was the extensive records and archives of the British museums of all the species that habitate the British Isles, i.e., there was an outstanding catalog for this project. And second, and perhaps most importantly, was focusing on native species, simplified sample collection, and removed import complexities introduced by the Nagoya protocols. Shortly thereafter, the European Geographical Initiative, ERGA, began to sequence all life living west of the Urals. For ERGA, Dresden has sequenced 23 fish comprising 26 billion base pairs of finished sequence. In summary, since we began in earnest in 2016, the Dresden Center has sequenced 79 species to the 2016 VGP standard, totaling 194 billion base pairs of finished sequence. And more recently, we've sequenced another 87 species to the 2020 VGP standard, totaling 108 gigabase pairs of finished sequence. Briefly, here's a list of the bats we've sequenced. About half are to the highest current standard with contig N50s over 10 megabase pairs, chromosome level scaffolding, and a 1 in 100,000 or better error rate in the finished consensus sequence. I'd also like to point at two of the first fish we sequenced for the ERGA project, noting in particular that these are haplotype phased assemblies. That is, for every chromosome, two sequences are produced, one for each haplotype of a diploid pair. Briefly observe also that the continuity and quality of the two haplotypes are comparable. So haplotype phased reconstructions are just now becoming available. Here on this slide, we want to look at an interesting revelation for the pearl oyster, only visible with such an assembly. The pearl oyster has now been sequenced 3 times by Takeshi Takeuchi in the Nari Sato lab. This last time with my lab as a collaborating partner. In the plot, you see the contig lengths plotted as a curve from the longest to the shortest contig. Each successive assembly with better technology brought about an order of magnitude in the contig length. In particular, note carefully that the hi-fi curve is of phased contigs, whereas the earlier curves were of consensus contigs. When these contigs are then scaffolded with high C, one essentially gets the genome in 28 large scaffolds with a sharp drop thereafter. The pearl oyster has 14 chromosomes, and so the 28 scaffolds represent the 14 pairs of haplotype sequences for the genome. The high-C heat map shown there visually confirms the correctness of the scaffolds. So what we did is we took each pair of chromosome scaffolds and aligned them against each other. For the largest chromosome at left, the two haplotypes are in near perfect synteny. When we looked at the third largest chromosome, shown in the middle, there are some inversions, shown in blue, between the haplotypes, but basically, again, the two haplotype chromosomes are syntenic. However, when we looked at the ninth largest chromosome, there were three large megabase pair regions where there was little synteny between the haplotypes. At first, we thought there was some kind of assembly error, but after thoroughly vetting the assembly in these regions, we were convinced that they are correct. So when we investigated the genome, what we saw was that there was a lot of evidence for very recent retrotransposon expansion, especially in this chromosome. So we speculate that this expansion may be mechanistically responsible for the large non-synchronous regions. Moreover, note that this lack of synteny implies that these portions of the genome are much less likely to recombine during meiosis, as sequence homology is required for recombination to occur. So these regions, we imagine, tend to stay fixed in the genome. So when we look into what types of genes are in these non-synchronous regions, we found them heavily enriched for genes involved in the innate immune response, especially NOD-like receptors. Moreover, the receptors were not synchronous between the regions, occurring in different locations and in different copy numbers with different effectors and recognition domains. So this leads us to hypothesize that because of these non-synchronous regions, the pearl oyster has an expanded immunity repertoire approaching twice what would occur in a synchronous genome. So inbreeding oysters has been notoriously difficult as the animals are almost always sickly. And so we speculate or suspect that it could be due to the fact that the inbreeding removes one of the non-synchronous haplotypes, thus reducing the immune repertoire of the inbred strain. So this is the first of two examples that demonstrate that there is potentially interesting genomic mechanisms that are only apparent when one sees the haplotype-phased genome. The second example is from the work of my good colleague Axel Meyer, who has extensively studied the cichlid fish that swim in a series of volcanic lakes in Nicaragua. One of the species presented are either black or gold in color, and a gene or mechanism explaining this had eluded detection for years until finally a haplotype-phased genome of a gold individual was produced. What they found is that in a gene called golden touch, an intron of one of the two haplotypes, but not both, had an 8.2 kb, 8200 base pair retrotransposon insertion, whose palindromic sequence forms a double hairpin that prevents
the transcription of the last exon of the gene. PCR probing of Black individuals revealed that this was only true for the gold individuals. It was then confirmed that the gold phenotype is the result of a half dosage of golden touch expression. So while these are only two examples, I think that these are not necessarily rarities, and that phasing should be a part of the standard for biodiversity genomics explorations. Even with HiFi, many genomes are still being reported as a consensus sequence, primarily because the continuity statistics look a bit better. However, I think that we should resist the temptation and start reporting the phased haplotypes aligned to each other. So indeed, we are almost at the level of producing contiguous T2T genomes, and the push is on to achieve this. Already in the seminal work of Nurk et al., this was achieved for the haplotypic mole CHM13 by threading the HiFi string graph with an excess of ultra-long nanopore reads. Applied to diploid genomes, this nearly works, but is not enough to resolve and separate the haplotypes everywhere. It is also a bit expensive to produce all the ultra-long reads. Ultimately, some form of long-range path information at an inexpensive price point is needed. Work is ongoing, and it's very likely only a matter of time before we get there. So in conclusion, projects such as the Vertebrate Genome Project to sequence all the vertebrates are gaining momentum and becoming more and more established. Affordable, high-quality telomere-to-telomere phased reconstructions of multiploid genomes are only a matter of time, in my opinion. And I think that it's very important to sequence and report haplotypes, not a consensus thereof. And so I'm very excited about where we're headed, because I think we're going to be able to begin to explore the genomes of the natural world and of the human genome in an unprecedented way. So I'd like to acknowledge and thank the many people I've worked with in the last half dozen years. Thank you very much. Thank you.